3 kinase enhancer, which is a nuclear GTPase in the nucleus. So that means that if EGF is a upstream signal targeting the PLC gamma in the nucleus by EGF receptor, then we can look for the downstream targets of these events. And at this stage, I was approached in 2002 by a student, Christian Klein. He desired to do his PhD, and so I gave Christian all these papers, and after a few months he appeared and he proposed that these experiments can be done in vivo in mice. And these are the results that we have come out of that. Now, here, you see, the mice, and of course in this work, the Christian was assisted by our long-time collaborators, Solange and Claire. You see, intraperitoneal injection of mice and isolating the nuclei at different time intervals of the injection, and then here is a Western blot. You can see that phospholipase C gamma, which is a 150 kilodalton protein, at 10 and 30 minutes, this is just a control that you can see that there are 2 proteins which we have identified after the EGF injection, 150 kilodalton and the second one is 120 kilodalton. So this was little puzzling, but when the same blot was immunoblotted with anti-phosphotyrosine antibody, what we see that the 120 kilodalton protein at 10 and 30 minutes after EGF injection were tyrosine phosphorylated, but not the 150 kilodalton protein. And that 120 kilodalton protein is tyrosine phosphorylated in the nucleus in response to the EGF was further confirmed when we immunoprecipitated this protein with anti-PLC gamma antibody and immunoblotted with anti-phosphotyrosine. You can see these two are indeed tyrosine phosphorylated. And so this gave an intriguing idea. Is 150 kilodalton and 120 kilodalton related with PLC gamma? And the answer to this question is here. We pre-incubated the antibody, anti-PLC gamma antibody, with a blocking peptide, and we see both 150 and 120 kilodalton proteins in the immunoblot disappear. Then where is the question of cytoplasm? What is happening in cytoplasm? And we can see that in cytoplasm, the PLC gamma remains intact at 150 kilodalton, and it is at 10 and 30 minutes EGF that it is phosphorylated. So cytoplasmic event seems to be a different event than what is happening in the nucleus. But then the question arose, what is the status in this mode of the EGF receptor? And Christian has identified EGF receptor present in the inner nuclear membrane of the mice, which is a 170 kilodalton protein. So here is the status of the immunoblot with anti-EGFR antibody. You can see 170 kilodalton under the similar treatment. Anti-EGFR and immunoblot, immunoprecipitated with anti-EGFR, immunoblotted with anti-active EGFR at tyrosine 1173 or tyrosine 1068, and you can see that the that EGF receptor is phosphorylated under these conditions. Now that means that EGF signaling to the nucleus, as shown by Cohen, is indeed stimulated in the nucleus. And this, then the question is, what is the relation between the EGF receptor and the 120 kilodalton protein, which we think at this time is a derived product from the nuclear native 150 kilodalton PLC gamma 1. And here is that when immunoprecipitate anti-EGFR, immunoblotted with anti-PLC gamma 1, you can see 120 kilodalton. Similarly here and here. And when immunoprecipitate with anti-PLC gamma 1 and immunoblotted with anti-active EGFR receptor, you can see a 170 kilodalton protein. So that shows that the two moiety, 170 kilodalton and 120 kilodalton, which we begin to think is probably a breakdown product of 150 kilodalton. They are these two moieties associate themselves upon both the moieties being stimulated under EGF signaling. Now, this raised the question, what are the downstream targets? Downstream targets of the EGF signaling. And the next shows, here it is. You see, the phosphatidylenoacetyl kinase enhancer protein, when immunoprecipitate of anti-pike, immunoblotted with anti-PLC gamma 1, what we can see here is the 120 kilodalton protein. And just converse, immunoprecipitate of anti-PLC gamma 1, immunoblotted with anti-pike, you can see here is the 98 kilodalton anti-pike moiety. Now, then the next question is, because pike, the other is the PI3 kinase. What is the status of PI3 kinase? And here is immunoprecipitate of anti-P110 antibody, immunoblotted with the anti-PLC gamma, you can see 120 kilodalton protein. That shows that pike and PI3 and PLC gamma, which is 120 kilodalton protein, they form a complex. And if they form a complex, then what is the natural? That there should be an enhanced PI3 kinase activity under the similar paradigm. And you can see here the PI3 kinase activity in 10 and 30 minutes treatment are enhanced. So this support that we have gone through from one stage to the other, and then there is a generation of increase in the activity of the PLC gamma 1, which generates, of course, diacylglycerol and IP3. Of course, there are other mechanisms of diacylglycerol generation, and that we propose that it is the mechanism by which the nuclear IP3 is generated. It is unfortunate that till today there is no way to determine the IP3 in the nucleus quantitatively. We think, and many others think in the same way, that IP3 is generated in the nucleus to mobilize the calcium, but does not stay long enough to be determined by the available techniques. And so the next, then we addressed the question of 120 kilodalton protein. And with the collaboration of B.C. Nair, we have been able to partial sequence 120 kilodalton protein at N terminal and both at C terminal. At N terminal, this is, first count is the proline 244 and glutamine 253. And this sequence is identical with the human and rat PLC gamma 1. So this gives us an idea that 120 kilodalton protein fragment represents PLC gamma 1 minus its N terminal 1 to 243 amino acids, and the site of cleavage is located between arginine 243 and proline 244 in the mouse liver PLC gamma 1. This is our current way of thinking. The next, please. Now, so we propose that the 120 kilodalton nuclear protein is derived from the cleavage of 150 kilodalton PLC gamma 1 native nuclear enzyme, and this is based on these facts. First, is immunoprecipitated with isozyme specific PLC gamma 1 antibody.